Welcome everyone to the new format of the Blue Brain Seminar Series. So this is the first online Blue Brain Seminar. And thanks to Maria Reva for her help in setting this up. And it's actually a pleasure to have Albert Gidon as the first speaker of our online Blue Brain Seminar Series. So Albert is a repeat Blue Brain offender. So he was one of the first members when Blue Brain actually started more than a decade ago, and he did his PhD at Iran Segev at the Hebrew University in Jerusalem. Did some interesting work on principles of inhibition in dendrites. And then switched gears to do an experimental postdoc with Matthew Larkham in Berlin, where he's been doing some very cool work in human neurons. So without much ado, let's hear about all the amazing work that Albert's been doing.
Well, hi, good morning, everyone. First of all, I want to tell you that it's a pleasure speaking in this platform with you. Two reasons that I feel at home now. One of them is because I'm really at home. The second is that because for a long time I was going back and forth between Switzerland and Israel with the project, with the Blue Brain project, was a lot of fun and pleasure. So, it is good to be back. And this is one of these projects that bared a very nice surprise when we started to do it. And I'm going to talk about this, the biophysical mechanisms underlying dendritic computation in the human cortex. So the outline of the talk is, so first I introduce the subject more or less, and then I'm going to talk about 3 lines of results. The first one is about the decoupling of the dendrites from the soma in the cells in the layer 2-3 pyramidal neurons in the human cortex. Second results, I'm going to tell you about this, as I mentioned, the nice surprise, a new dendritic spike that we discovered in the human cells. And the third result is about the computation in these dendrites due to this spike. Then I'm going to talk about the model, which probably will interest some of you. So we all know and we all discuss the fact that the human cortex is bigger and thicker, and we all saw this slide about the progression of the thickening of the cortex during the evolution from insectivores into humans. But we also know that the thickening of the cortex is not uniform. What we see is if you do a comparison, if you look at the y-axis, this is the layers and this is the thickness of the layers in the cortex. And the different bars are the mouse and the rat and the human. You can see that the majority of the thickening of the cortex in absolute terms is due to layer 2-3. And this is no accident. And there is fantastic work about the thickening of the cortex in the last decade in humans and in primates. And there is there are several aspects very unique to humans and primates in the thickening of the cortex. And why is that interesting? Because this thickening is attributed mostly to layer 2-3, and it is also in the development. So that's kind of interesting. That's prompted us to have a look at layer 2-3 rather than layer 5, which we used to do in the lab Matthew lab used to do for many years. So as you all know, that the thickening of the human cortex is also resulting in large dendrites. And this is a comparison. I'm not going to do a lot of comparison here between mice and humans, but this is just one of a few slides. So you can see the differences, and this is a human layer 2-3 cell, so it's quite big. So it extends up to 1.2 millimeters, and the dendrites are pretty long. And besides the fact that that's really beautiful dendrite, and you, when you see it, you really appreciate. You see also the spines, and this is really nice. Beyond that, there is an issue that really begs a question about the interaction of the very distal synapses with the soma, because we know that as you go distally, there is an attenuation of the signal, attenuation of the potential from the very distal sides. So there is a question, what's going on there in terms of interaction? Right? And so the physical dimensions is not everything. We all know that you need to look into the electrotonic size of the dendrite. So it could be that the dendrite is very compact and the large size is really not an issue. It's not an issue in the sense that the signal or the input from the very distal synapses actually has the same effect at the soma as it has locally, which means that the cell is really compact electrically. But it also could be that the cell is very large electrotonically, which essentially means a huge attenuation of the signal from of the inputs from the distal sides to the proximal sides. So the fact that we have big dendrite doesn't tell us too much about the attenuation of the input to these distal sides. Why is it important? Because when you have an apical dendrite like this, what you have is that the majority of the inputs are very distal. They are coming actually from the area of layer 1 at this apical dendrite. So it is important to tell what's going on in terms of interaction of these distal sinuses with the soma. So here is a hint that came out from Idan Segev lab in 2017. And they did measurements of the apical dendrite of layer 2-3 cells, and the entire dendrite, in this case, the apical dendrite. And here they measured the surface area of the human apical dendrite in layer 2-3 versus the mouse apical dendrite in layer 2-3. And you can see very obviously that the human total surface area is much larger than the mouse. Now they went one step further and they looked at the diameter. And that is astonishing to see that the diameter is the same. And why is that? Why is it astonishing to see that? Well, that's make my life as going to the dendrite and trying to record from the dendrite very hard. So initially, when we started the project, we saw that it's going to be a very easy project because the dendrites are so big and so thick. But no, that's not the reality. But the second thing is that if you know anything about attenuation of potential from cable theory, you know that if you want to make the dendrite more compact, what you need to do, the first and easy step is to make the diameter of the dendrite just a bit bigger, and that will help to compacticize this dendrite. And it doesn't happen. And that's the first clue that something is very fishy. Something very fishy is going on here. So we went to do the experiments, and we get the tissue from a patient that have a temporal lobe epilepsy. Most of the cases, we have a little bit of tumor patients as well for controls, but that's the majority of the tissue. And the epilepsy is coming from patients in different ages, epilepsy onset, different sex, different history of drug use for this disease, seizure frequencies, etc., etc. And most of the cases, temporal lobe epilepsy involve epilepsy focus in the hippocampus. So what we get is essentially tissue which is resected to have better access to the focus of epilepsy, to remove it. But this is considered to be a healthy tissue most of the times. We don't know for sure, right? Because you can never know for sure, but the assessment is that you're talking about a healthy tissue. So we basically take the tissue from the surgery. It used to be half an hour. Now it's like 10 minutes away from the lab. And we just put it on ice, take it and process it. So we remove the pia, we slice it about 350, 400 micrometer slice. And there is some, I mean, it doesn't work like the mouse, where you know exactly how you slice it. So there is a little bit of artistic touch to slicing this human tissue. And then we do the electrophysiology. And so I'm going to show you the first results that we get. So mostly what we do is dual recordings. In this case, you see 3 pipettes because that's 2 consequent dual recordings. So we start with the soma, we put Alexa at the soma so we can visualize the dendrite, then we patch the dendrite. And so these are 2 electrodes in the dendrite, stimulation at the soma. You see that the current here, step current, very simple stuff. and then you evoke action potential at the soma. Then you see the backpropagating action potential in the dendrite, 180 micrometers from the soma, attenuated version. And then we do it again, and you see that that's what you get as the action potentials at 425 micrometers. And you can see the attenuation of the backpropagating action potential. It's really, and this is a zoom in for one of the spikes, really massive attenuation. That came as a surprise. That came as a real surprise. But this is just one case, and we did it for something like 50 cells. And you see that the picture is exactly the same. There is electrical decoupling of the apical tuft from the soma with respect to the backpropagating action potential. And the lambda that you get, the lambda is the space constant, is about 300 micrometers. So if you remember that the size of this dendrite is about 1.2 millimeters, and the lambda is 300 micrometers. So you have about a dendrite, which is about 4 lambda long, which is a huge attenuation of the signal, which make it essentially non-existent in the very distal dendrite. And if you do the same, so if you injected the dendrite and you measure at the soma, you see, so here you inject now in the dendrite and you measure the amplitude at the soma, you see the same picture. There is a massive attenuation of signals that come from the very distal dendrites to the soma, and that is strange. That is strange. If there is such a massive attenuation, why do you need these very distal synapses? How does it work? There is something here is missing. So I'm not going to talk too much about this, but we did also 2 photon imaging to look at the calcium signal, and the picture is very similar to what we see with the potential. There is a frequency dependence here, but still you get a massive attenuation in very distal dendrites. And that means that spike time independent plasticity is probably doesn't do anything in the very distal dendrites with such a steep attenuation. So how do you get the information of the somatic spike so distally? That's unclear now. But then you'll tell me, wait. So that is what you see here. Oops. What you see here is the model that we used to have, or we have now for the rodent layer 2-3 dendrite, which is a very compact dendrite. So you have the soma, and then attached to it the dendrite, the basal and the apical dendrite. And you don't basically need more than that if you talk about a small compact cell. But this is the current model because of this attenuation. Do you see also the mouse instead of me pushing all of these buttons all the time? Do you see the mouse?
Yeah.
Actually can't. Do you see the mouse on my screen?
Yes.
This is a mouse.
Yes. It's visible.
Okay. So this is a model now of the cell in the human cortex, a layer 2-3 cell in the human cortex, and we see a very strong decoupling of the apical dendrite from the soma and the basal dendrite. So that would be the model that you're going to use when you talk about layer 2-3 neuron in the human pyramidal, in the human cortex. But then you'll tell me, look, we know that that's not the entire story, because we know that in layer 5 in rodents, if you record at the dendrite, what you're going to see is a boosting of the inputs at the apical dendrite with the calcium action potential. And not only that this input can trigger the calcium action potential, this calcium action potential also can trigger a burst at the soma, as you can see here. This is injection in the electrode and measurement in both electrodes at the soma, so you can see the calcium spike, and then you can see the burst. Can be 2 spikes, can be 3 spikes. And so they have in layer 5, there is a mechanism that allows the boosting of the very distal synapses. We don't know of something like this that happens in layer 2-3. So this is the model for layer 5. And you see here the area with the calcium spike, and the NMDA at the tuft, and then that one travels to the soma and activate. That is the current hypothesis, that this boosting is required in order to have the effect on the output of the cell. In layer 2-3, we think we don't need that, right? That's the way we see it. We don't need that because the cell is pretty compact, but the reality of, in the human case, the human layer 2-3, that the cells are quite big. So what's going on? That's what I want to show you the second result. So we did the second basic thing, and now we stimulate the dendrite with the current pulse, and now measuring both dendrite and the soma. And this is what you get. You inject at the dendrite now. This is the current pulse at the dendrite. And now what you see is spikes at the soma, action potential at the soma. And then you see what we thought was backpropagating action potentials in the dendrite, which seems like quite a trivial picture until you look at until you zoom into these spikes. And then you see that the spikes at the dendrite precede the spike at the soma. And that was very nice surprise to see because that wasn't expected. And you can see that well, I wrote here dCaAP, which is an acronym for dendritic calcium action potentials. And we know, initially we thought that was a sodium spike because it was relatively thin. But then we tried to block. So we saw that with the blocking experiments that these spikes are sensitive to cadmium, and they are not sensitive to TTX. And the initial thought was, well, maybe that is a different sodium channel, which is not sensitive to TTX. And then we replaced the ion in the bath, the sodium ion in the bath, with NMDG, and you still get these spikes. So these spikes is probably, I would say even most definitely, calcium spikes. And they are thin calcium spikes in these layer 2-3 cells. So just for comparison, if we do a similar experiment, only now we inject current at the soma, we see the spikes at the soma, and then we see now we think the backpropagating action potentials at the dendrite. And when we zoom in, that is definitely the somatic spike precedes the dendritic spike, and that would be what we commonly see as the backpropagating action potential. Now, if you look at these two spikes, that's an average spike, but you see individual examples in the background in gray. So you see that the anatomy of the dCaAP is different from the anatomy of the backpropagating action potential. In several ways, it's a bit thicker. It's not as thick as layer 5 pyramidal, which would be about 60 milliseconds and more. This one is about 4 or 5 milliseconds half width. And you see that it's very slow rise compared to the backpropagating action potential, which has this kink right here, deflection point right here. And when you look at these spikes individually, it's very easy to say when you see backpropagating action potential and when you see dCaAP in the dendrite. Right? It's not a big mystery. You don't have to do any analysis. You just see, exactly what you see once you know what to look for. So interestingly, we have a dendrite that has two distinct fast action potentials. One is the calcium action potential. The other one is the sodium backpropagating action potential. And it is not clear to us what is the interaction now between these two spikes. Right? That's an unanswered question yet, but it exists in both of these. We have preliminary ideas and preliminary results, but it's not clear what the interaction is. This is still something to look at. So now we classified the. So what we see essentially is that we have a lot of different spikes. We have this per 12 action potentials in the dendrite. So we decided to classify them to see if we can get some handle to the different kinds that we see. And the first one I'll show you is a coupled simple dCaAP, which is essentially every time that you have a spike at the dendrite, you will also have accompanying spike at the soma. So this dendritic spike, seemingly at least, causes a somatic spike. And what we see always in the case, in all the recordings that we have, is that one spike at the dendrite causes one spike at the soma. It never came as a burst as in layer 5 pyramidal cells. And if you want to look carefully, you can see that these spikes are time-locked very nicely, and the dendritic one precedes the somatic one. The second type is the coupled but complex dendritic calcium action potential. And what you see here is a bit confusing, but I'll explain. So the first one here, the first spike here and here, is a dCaAP. And the second one is the backpropagating action potential. So it's not a burst of two action potentials at the dendrite or at the soma. One at the soma and one at the dendrite. And you can see if you zoom in, you can see what happens. And that one is a strange thing. We thought initially that was just by chance, but apparently it's not. It's something very consistent happens. So you have a spike. So we inject again at the dendrite. What you have is a spike at the soma. This red spike at the dendrite, and then you have a wiggle. It's like a wiggling of the membrane potential. And then you can see the backpropagating action potential in the dendrite. At the soma, the picture is reversed. You see this blip here at the soma, and then you have a ramp up of the membrane potential until you get to a spike. And then you get a hyperpolarization of the spike. And this is very consistent. You see that we saw that several times, and that's very interesting because it means essentially that you can have delayed spike. You can, if you can control it, if you can control this delay, if the cell can control this delay, and we don't know that it can. We don't know that. But if it can, the cell can engineer all sorts of coincidences or to avoid coincidences of activation of different pathways. That could make that very interesting, but still need to be shown that the cell can really control something like that. So there is an inherent way to control it that we are not aware of. And then the last one that we classified would be the uncoupled and the simple. The simple is because we don't have this complex activity as I showed you in the previous slide, and the uncoupled is simply because when you have spikes at the dendrite, you do not see corresponding spikes at the soma. Right, that's right, this one, this is a simple one. So the third result that I'm going to show you. So usually when you work with human tissue, it seems as if there is some kind of a glass ceiling, because you never can take that forward to do in vivo work. And that in a way is kind of problematic if you want to understand function. But I'll show you that there might be ways to get some understanding of function even if you don't have access to in vivo. At least it can inspire then when you have access to something similar, let's say in primates, then you can examine these things further on. So what's the function of this dendritic calcium action potential? So obviously, we don't know the entire story, but here is something. So the arrangement of the experiment is that we inject current at the dendrite this time, and we record both at the dendrite and at the soma, and this is progressively increasing amplitude of step at the of current at the dendrite. Right, this is the dendritic current. And what happens is that in the first step, in this case, specifically for this case, in the first case, there is a somatic depolarization, but you don't see anything more than that. And then somatic depolarization, you get the first spike, and then you increase the current to the dendrite, and you see two spikes, and then you further increase the current, and then you see no spikes. And then you further increase the current and you see no spikes. And that is a very strange situation. Yes, well, it could be a random thing, right? It could be by just noise. But it also, if you increase the current, you usually used to the case where you get more spikes and not less spikes. And that was strange. But now we can look at the dendrite and see why, if there is a dendritic basis for this behavior. So again, we look at the same. So this recording here corresponds to this recording here. So this is a dendritic recording, this is a somatic recording, and these two correspond to this current pulse here, right? So now this is the first amplitude of current, and we didn't get anything in the dendrite. Now we increase the amplitude and we get a spike in the dendrite. And this spike corresponds to this spike here. And now increase the current even further and we get 2 spikes at the dendrite. And these 2 spikes cause 2 spikes in the soma. And now we increase the current. We see that the spikes became smaller, decrease in the size of the action potential. And this wasn't enough to trigger the spikes at the soma. And then we further increase and we see there is a little bit bigger here, but still no spikes here. So we didn't see anything at the soma as well. So and just to be clear, every spike that you see here has the same feature of preceding the somatic spike. Right? So it's very clear to us that every one of these spikes actually caused or contributed, however you would like to see, to the spike at the soma. And this one just didn't make it, even when we increased the current pulse at the dendrite. So this captured our eyes in terms of this is a very nonlinear way for an input-output function of the cell, and that can be very interesting to look at further. So what we did is now we inject current in the dendrite, progressively increasing current in the dendrite, and we measure the amplitude of the dCaAP. Right, that would be the amplitude of the dCaAP as you increase the current at the dendrite. And what you see here is that's the amplitude, and this is the current intensity to the dendrite, and every line here is a different cell, right, showing the different progression of the height of the action potential in the dendrite. So you get a decrease in the action potential in the dendrite. And now we wanted to have some kind of an idea of what would be the average effect, right? The average effect. So we combined all the data together. We normalized the amplitude to the maximum, and then that would be the first spike in the series because it is usually the maximal one. And then we normalize the input current in the dendrite to the raw base, to the dendritic raw base, and you get this curve that tells you what is the sensitivity of the dendritic spike height to the strength of the input. Right? And what we can see, and you can characterize this curve by a tau, by this dCaAP constant, and the unit will be of this constant of raw base, right? It's in units of raw base. And what you see is that the dCaAP amplitudes are sharply tuned to the optimal input intensity, and then they decrease very rapidly. So if you do the same thing at the soma, like we know that also sodium action potentials get inactivated. So maybe it's not such a big deal. We do the same thing at the soma. We want to see how much the soma is sensitive to the increase in the current pulse. And what you see is that it's virtually in a reasonable range. It's independent of the input intensity. So you increase the stimulus and you see that there is no reduction in the height of the spike. And if you do the same kind of analysis, you normalize the raw base versus normalize the amplitude of the spike, you see that there is almost no reduction in the height of the spike. Of course, that will break down if you increase the input, but in the range of activity, that's very, very stable. And we also know that for other dendritic spikes, except the somatic spike, NMDA spikes and the calcium spikes, so they will, initially at least, they will tend to increase with the increased input, not decrease. Right? The size of the NMDA spikes will increase as the more synapses you add. And the size of the calcium spike doesn't increase, it will remain pretty constant with increasing input. So this is a feature in the cortex, at least. This is a unique feature for this spike, for this dendritic spike. So let's update the model now. And if we want to characterize, the different compartments in the cell, we can think of activation functions, which is virtually the input-output function of this compartment. And now we can divide the cell as having this different activation function. The soma one is the step, right? Or if you want to, you look at the the rate, then you can think of a IF curve. But in terms of the size of the potential, you get this step function. And at the basal and the apical dendrites, you get this sigmoidal NMDA activation function. And at the apical dendrite, that's a new function. You get this tuning curve-like function, tuning curve-like activation function in the apical dendrite. So we have all this kind of activation functions that can interact together and can make this neuron very interesting in terms of computation. So let's get to the model. So the immediate outcome of putting this model together was the following. So we have this reconstruction of layer 2-3, one of our layer 2-3 cells, and you have engraved the background synapses. And in the different colors, you have different pathways. So the red one will be pathway X, right? So there are inputs that are activated more or less at the same time. And then you have pathway Y. It's a different pathway. It's the same area. And simply pathway is a group of synapses that have a common activation. And then you have the yellow ones, which inhibitory synapses. And now here, the calcium action potential, the dendritic calcium action potential, is modeled by a phenomenological model, which virtually means that there is no biophysical mechanism behind it. And the reason is that we didn't want to guess anything that we don't know. So we can build a biophysical mechanism as we did for this work. But for that particular example, we didn't want to assume more than we know. And what we know is that at the point of measurement that we see these spikes. And that's what we wanted to assume, no more than that, and that's what we wanted to examine. So you see the location of this dendritic action potential here, where the electrode is measuring all the traces that I'm going to show you now. So the synapses are activated in the background, and then you have on top of that pathway X activated, and it's sufficiently strong to generate this dCaAPs in the dendrite. And the same thing happens when you do pathway Y. You get these dCaAPs in the dendrite. And once you activate either pathway X or Y, you get the activity. But when you activate both of them at the same time, you see a decrease in the amplitude of the spikes. And that is exactly the kind of function you expect from anti-coincidence function. Right? You get a decrease in activity when you have these two pathways coming together. And this is a natural result of this tuning curve, right? This XOR-like behavior. What's interesting when you start to introduce these nonlinearities into the dendrite is that now inhibition is doing something which is completely not intuitive, right? We add inhibition in this case, and we see more excitation. Right? We see more excitation, and let's update the model now. So I think when I showed that to a group of modelers, that could be a bit annoying because this is the gross simplification of the function of the dendrite, and the intention is not to say that it is simple, but the intention is to have a take-home message which is clear and capture the differences that what we learned in this study. So what we have is essentially at the soma we'll have something like an AND or an OR gate, right? Depending on how you look at it and what the input that get into the soma, and this will be the activation function. At the basal and apical dendrite, you'll have this AND gate because what you need is cooperation of different inputs in order to get this NMDA spike. Again, in the apical tuft and the basal dendrites. And then at the apical dendrite, you'll have a XOR logic gate or XOR function, as I showed you in the model before. And that's it. So here's a summary of what I talked about. So the apical dendrite of layer 2-3 neuron in human cortex are electrically decoupled from the soma. And for me, this result tell me that we don't really understand what the cells are doing. That was because if what these cells are doing are integrate and fire, right? They just integrate the input and essentially reach a threshold and fire. That is the main thing. You do not decouple any part of the dendrite. You want to hear, you want to listen to all the synapses coming from different areas of, or collecting inputs from axons that coming from different regions of the brain. You don't want to decouple these synapses from the output. And then we have this dendritic calcium action potential, which have a lot of consequences for many things. I mean, we can think of plasticity, right? Now, how plasticity behave in face of this dendrite, of this spike, of this dendritic spike. We can think of now we have all this nonlinearity in nonlinearities in the dendrite, which include the NMDA and the backprogradient action potential and the dCaAP. So how do they interact together, right? Suddenly things become very interesting. And we don't know that either, how these things interact, how these things affect the learning of the neurons. What is the learning mechanism, given that you have this spike now? Third point is that these dendrites have dendritic action potentials which are sharply tuned to the input intensity, and that could be interesting. That could have interesting computational outcome, like the anti-coincidence detection that these cells can do. And as I said, so I've been doing for some time modeling and looking at dendrite, and this one really convinced me that we are scratching the surface of what these dendrites are actually doing, and we have to look in experiment and in modeling to try to understand what kind of interesting things we don't understand about these cells and what they're doing. Okay, so I'd like to thank the people who did this work. So the anatomy of these cells was done by Imre Vida and Felix Baldwin. Very good work they did, very, very precise with the anatomy. It was really nice to work with them on that. Tim Zolnik did the 2-photon imaging, which you just see a glimpse of. And we get the neurologist from the Epilepsy Centrum and the neurosurgeon Pavel Fitzinski, Martin Holtkamp, and Ulf Schneider, which provided the tissue, and that was not easy thing to do, but we get the tissue very nicely. And then modeling was done with Yota Poirazi and Nasi Papuzzi, which was a lot of fun to do this modeling. That was interesting, and you just saw a very little part of it. And of course, to Matthew Larkham, which is a great scientist, and a lot of pleasure to work with on this project. That was a lot of fun. So thank you very much for the time and for listening. And I hope that wasn't too long in these very strange times, and thank you.
Right. So yeah, thank you, Albert. Unfortunately, to disappoint you with this format, we cannot have a resounding round of applause, but you virtual clap instead. So now, people have any questions? So can you please use the raise your hand feature and unmute yourself, please?
So hi.
I have a question.
I have a question. Can you hear me?
Yes.
Okay.
It's a wonderful piece of work. And of course, it doesn't make the dendrites much simpler. But what I find even more complicated is that your dendrites were actually having multiple apical dendrites. Mm-hmm. So how do they interact with each other?
Oh, that's a wonderful question, of course. So I'll just give you a hint that things are as complex as you think in that sense, because in many of the cases, the apical dendrite bifurcate relatively early. And we have the chance to record only from one branch, and we see the spikes. So there is no good reason to assume that it exists only on one branch and not the other one. Right? There is some kind of symmetry that you expect from these cells. Right? So you will find them in one area, or one branch or the other one. That's one hint. The other hint is that whenever we patch when we patch the dendrite, we look at backpropaganation action potential, there is a clear decrease of the height with the distance. But when we do that for the dCaAP, we don't see that there is a decrease or increase with the distance. So they are distance independent, which gives another hint that they are all over the place. Again, we don't know that. We don't know that for sure, but it's a hint that they are all over the place. So the kind of interaction that you get among them with the synaptic inputs, with NMDA spike, this is something that we are currently working on. We're trying to understand that because that's a fascinating subject.
Yeah. Just to follow up on this, dendrites come in bundles. You know that?
Uh-huh.
Mm-hmm.
That's because
and
of
so
development.
One question which I've had for a long time, because we discovered some principles of these bundles, is how the dendrites which are sitting in the same bundle interact with each other. Is there any kind of electrical coupling between?
No, so I think there are people here that could answer this question, but I think for these specific neurons, I don't think we have an answer. Layer 3 neurons, I don't think there is electrical coupling in the dendrite of the cell. It's not widespread one. It's unlike the inhibitory cell side. It's not widespread if it exists. But in the human dendrite, we don't have data. We have some groups that did multiple recording for multiple cells. Right? Like your Geiger, they did multiple recordings, like multi-patch in human tissue. I don't think that they say anything about the coupling, the electrical coupling.
Mm-hmm. Mm-hmm.
It probably could be better answer, more elaborated on that.
Yeah.
Right.
Thanks.
On to the next question. Felix has a question.
Albert, wonderful talk. Thank you so much. It's a real pleasure. I have two questions. One is, is it obvious to you? You're saying that these dCaAP spikes, you worked out the biochemistry that sort of, it's not the usual ions. They're very fast, right? Is it clear to you sort of, which calcium channels sort of can create such fast spikes? I find it it's so different from other calcium signals we're seeing. So are you really convinced that this is just calcium?
Yeah, well, that's the way I treated this question for a long time. Am I really convinced? Right? And it took some time to be convinced that these are calcium spikes. And so why am I convinced? The rise time of this spike is not that fast, and it is comparable to what you see in other calcium channels. What's interesting is the fast decay. Right? So you have a slow rise, which is interesting in itself, right? So how you can have a slow rise and fast decay? How can you do that? Apparently, it's not such a big deal. You can do that with two channels. You can use a BK channel and calcium channel and get that done. So we have done this. But the rise time is characteristic of calcium, and then you have a strong inactivation or a strong decay of the spike, and that would be also fine. But now, for me, the proof was that if you just remove sodium ions, just remove them from the bath, you see the spikes. And without sodium ions, you cannot have sodium spikes, right? So what's left? Is it zinc? Maybe, I don't know. Probably not. That would be calcium. And we also know that as I mentioned, with the high frequency of backpropagation action potential, you might see something like that as well. So we did some experiments. Well, so you see you see increase in elevation of concentration of calcium when you have high frequency somatic firing. I think that might be due to these spikes. We didn't see anything else but these spikes. So given that you can block they are not blocked with TTX, they are not blocked with NMDG, they are blocked with cadmium. Yeah, what's left? Right. Now, what you want is the actual channels. This is something that we're working on, of course. That's the interesting bit, and eventually we'll get them. So if not someone else before us, but that's the best answer I can give.
And the second question I had, if I may, obviously, we've been so excited about the calcium hotspot in layer 5 pyramidal cells for rodents, and sort of, do you think that well, it's a speculative question. I mean, do you think there are cells in human cortex that sort of have that, or is that sort of different evolutionary paths that sort of in rodents, they sort of figured essentially they didn't get the XOR right, so they essentially just got the OR function, and then humans got the XOR. So how do you place this other cool mechanism of coupling distant dendrites? I mean, do you think you would find that in different cells or in the human, or is that sort of an evolutionary divide?
I do not think that this is an evolutionary divide, and here's why. So first of all, layer 5 cells, there is a study that published at the beginning of last year by Mark Arnett, and that was about layer 5 cells and recording of the dendrite. And basically, he finds that these spikes that we see in rodent doesn't exist in human, and so that is a result. But not seeing something doesn't mean that it's not there, right? So I think we will. I have good reason to believe that we will find evidence that the spikes are there, at least in some of the cases. Maybe the composition of the layers in layer 5 human is not the same as in rodents. But still, this kind of broad calcium spike still might be there. I don't think that's the end of the story. And that is to tell you essentially that layer 5 in human is layer 5 in rodent. And there is no evolutionary replacement of them. There could be some changes, right? It doesn't have to be the same. But layer 5 is layer 5. When you go to and you record from layer 5 rodent and you record in layer 5 dendrites, you basically see very similar characteristics. Most than anything, the IH, critical frequency, the elevated potential in the dendrites, they're all very similar in layer 5. And the same thing in layer 2-3. When you record layer 2-3 human and layer 2-3 rodent, they are layer 2-3. There is no mistake about that. There is difference in the size. There are some other differences in the spike width, in some other parameters, but they are layer 2-3 cells, and that's very clear. So there is no confusion of replacing layer 2-3 with layer 5, at least in the anatomical physiological sense. There could be some connectivity issue, but there are distinct layers, and it's very clear in rodents and humans. Now, about what's unique in humans. So that also has to be clear, because when you go back now to rodents, and we try to look the same spikes that we see in humans in the rodent, we do see something. It's not completely devoid of these spikes, that's right. And what we see is that the spikes exist in rodents, but it is not as evolved as in humans, right? So we can see this dCaAP, and at most what you get in rodent is one spike at the beginning of the stimulus. So it can exist, but is not as evolved. Repetitive action potential does exist. There is something additional that happens in the human, right, which prompts us to have a look at the spikes. But I think that there is some evidence that there is some form of that exists in rodent. But it could be that we are talking about two distinct channels, right, two different channels, maybe subgroup of the channels. So there could be a divide. And there are good reasons to believe why there are all sorts of divides in evolution between these different cells. So that is an interesting issue to look at these spikes. And if they are important, they definitely are doing better job in humans than in rodents, because they are much more developed. And we have also other evidence to show that these spikes are much more evolved in humans than in rodents, right? And I didn't have the time to present it here. So, yeah.
Wonderful. Thank you so much.
Any other questions?
Yeah, I have a question. So thank you for the excellent talk, first of all. And also it's more like a question/comment. So there is this work from the 90s. It was done by Zador and Brown, if I remember correctly, where they have no experimental data whatsoever, but they speculate how XOR problem can be solved. And they do it based on, I think, hippocampal CA1. So, and they make their dendrites, so-called cold spot, which is practically a patch of calcium channels or inhibitory calcium-dependent potassium channels. And they're able to reproduce the phenomena that you observe. So I think it's like interesting crosstalk with what was found back then, was thought to be in charge of the nonlinearities and XOR problem. Do you think there's like some connection on that level? And also in hippocampal neurons, so what's the sort of problem there, and what do you think about that?
Well, here's how I see it. The XOR is an immediate handle to look at the complexity that you have in the dendrite. But so the dendrites are definitely capable of solving the XOR problem because they have nonlinearities in the dendrite. And that thing, although it surprised the computer science community a lot, that was a big surprise there. For me as a biologist, and I think for all of us, it's not such a big surprise that the dendrites are complicated and they are capable of doing these nonlinearities. So whether the dendrites can do XOR, yes, they can do that, and they can do a whole bunch of other things that we would like to know about. And yeah, so I remember having a look at this paper. I don't remember right now, so I cannot exactly relate to what you said in the details. But yes, I mean, definitely.
It's just great that the calcium channels idea that they can be responsible for XOR. It was there for a bit. But what do you think it also brings some, I don't know, new paths for neuromodulation?
Neuromodulation.
Yeah.
I mean, in the sense of the coupling or the decoupling of the cell, this is one thing that is currently investigated at Matthew's lab. And so it is possible that we have coupling, stronger coupling with some modulation of the dendrite. But what's astonishing is that, remember that this examination of the decoupling of the dendrite from the soma is in vitro decoupling. So on one hand, you can say, well, you can modulate that in vivo. But on the other hand, once you have synaptic activity and you have all these conductances on the dendrite, now the electrical size of the neuron even increased, right? So this is another type of modulation that you need to think about, because now it will be even bigger and the distal synapses will be even more distant electrically from the cell body, less effective. So what's going on here, right? So that's even a bigger question, I think.
I have a short question, sorry.
Okay, go ahead.
Unless it's embarrassing because you're at home, I'll be happy if you show your face, because that gives when you ask a question.
Oh, I'm sorry. I mean, I just forgot to turn on my video. Sorry.
Yes, we all like now.
My question is that how sure are you that the tissue which you're recording from is not epileptogenic? Because the tissue which is resected is the one which is causing seizure, right?
All right. So there are several lines of reasoning and evidence why it's not an issue. The first one is that every layer 2-3 cell that we recorded from had these spikes in every subject that we examined. And this subject has completely different histories. So it could be that the disease affected the spikes. That's possible. But the fact that it's there I don't think that's part of the disease for that reason. Now, the second thing, as I told you, when we go back to rodents, we see something is there, and probably these rodents are not epileptic. And the third point, and I think that's the most convincing part, is that we also have one before that. So as I told you initially, the tissue is considered to be healthy tissue in most of the cases. So we know when the tissue considered to be sick, which is minority of the cases, and the majority of the cases considered to be healthy, right? So that's why we think it's not epileptogenetic, right? And before the surgery, the patients become drugs free, so they stop taking the drugs before the surgery. So we know that it's not, they may have long-term effect, but at least it's not in the system. And the last point, which is, I said, is the most convincing one, is that we also use tumor patients' tissue. And we have about, I think, something like 4 cells, all of them showing these spikes. So they definitely exist. It's not an issue of the epilepsy.
Okay, thank you.
Well, I have a couple of questions / ideas for follow-up experiments. So what if you were to redo some of these experiments in an extracellular medium with low calcium and high potassium? Then how would you expect this coupling to change? And another related question, which was already raised before, was with neuromodulation. So there was a recent paper by Stephen Williams where he actually showed that activating cholinergic release makes dendrites of mouse layer 5 pyramidal cells burstier, so it makes them more excitable. So do you see something similar in your experiments with layer 2
the last
cells?
part. Can you repeat the last part?
So Stephen actually showed that optogenetic activation of acetylcholine actually makes apical dendrites of mouse layer 5 pyramidal cells more excitable or bursty. So do you see something similar or contradictory in your experiments on the role of neuromodulation in dendritic excitability in human cells?
So we didn't do any neuromodulation. This is definitely something to do. Right? We didn't try to modulate the spike except of blocking them.
Okay.
Although there was one attempt to block potassium channels, and if I remember, that was a while back, and we didn't follow up too much on that because the results are not. Once you block a channel, you sometimes can get very dirty outcome, and you might. And I don't. If I see something dirty, I don't trust it. But the idea was, the thing was that the coupling was stronger. But that's not a big deal because the point is that if you do a passive model of this dendrite, that might be interesting. So if you do a passive model of the dendrite, you don't see the same attenuation. Right? In order to get the same attenuation with the passive model, that you'll have to really make an effort with the axial resistance to get this attenuation. So there is an active component to the attenuation of the potential, right? So what you can take from that is that it's not only that the dendrites are big, it's not only that the branches are thin. There is also active suppression of the signals from the distal areas, which might be because you want to modulate them, but it also might be because there is some other reason. So I don't have a direct answer to your question, but...
Okay. So do we have more questions for Albert?
I had like few very small questions. So in other type of primates, is there anything like that was observed before experimentally?
Oh, wow. So this is like, we have, the way I say it, we have a few small pieces of a very big puzzle that we don't understand about other animals, about mammals' brains. And we virtually have humans and rodents. We have a little bit of monkeys, but dendritic recordings of these cells, we don't have them. And we have some dendritic recordings from layer 1 in monkeys, but that's not very informative. So we are missing a lot of information. And if you want to think of divide in terms of evolution, what happens with the dendrites and which case development, that's impossible question to answer these days. And you can look at anatomy and maybe confer from that something, but that's it. That's unfortunate. So we had in mind for some times to do other mammals as well, but that seems to be difficult. What's strange is to get human tissue is easier in many aspects than get tissue from other animals, right? Because you have a guy who agreed to do that, and that's.
Albert, I was curious. I mean, you made this strong statement that this would say layer 2 in human clearly is layer 2 in it's layer 2. I mean, as sort of layer 2 in rodent, and layer 5 stays layer 5. But something that intrigues me is, I mean, one of the aspects of layer 5 pyramidals in rodents is the fact that they make it all the way up to the pyramid. And layer 5 pyramidal cells in human don't seem to be having sort of gone all that extension in size. So how can you keep up a statement that sort of layer 5 is layer 5, given that sort of layer 5 pyramidal cells in humans don't seem to be as prominently integrating all the layers? So I mean, somehow that's a little bit at odds to sort of what I heard before.
Yeah. So what you say come from a gallery of cells that was published by Christian de Kock, right?
Mm-hmm.
And
Well,
then
Javier says the same thing, that he says, look, they just don't make it up all the way to here.
well, so if you look at the study by Mark Arnett, you'll see that they do. And they do see the dendrites go all the way up. Maybe it's not all the dendrites. As I tell you, the composition of the dendrite might be different, but they do go all the way up. And you do see very nice apical tuft. Although in terms of branching, layer 2-3 looks like it's more branching more nicely than layer 5, but still you get apical dendrites that go all the way up. Maybe not all the time, but you do get it. That's for sure.
Mm-hmm.
Yeah.
Okay.
So. Yeah. But what I meant is, so you get that with the anatomy, and the anatomy is different. But when you go to the physiology, and you record from a cell, and you take any physiologist, and I'll show them the data, and that they did layer 2-3 and layer 5, they will immediately tell you, well, that's obviously layer 5, and that's obviously layer 2-3 by the physiology of the cells, by the recordings, right? So that is the strong where the strong claim is, right? Then there are some weaker claims that we can add to that.
Mm-hmm. Then I guess another follow-up question here is, so there was a paper in the Journal of Neurophysiology some years ago by Haddox and Sasha Nelson, where they showed that in the prefrontal cortex in rats, dendritic physiology is somehow regulated of sorts or dictated of sorts by the projection patterns of the axon. And now that we are talking about layer 2-3 cells and layer 5 cells, I mean, at least in juvenile rat somatosensory cortex, layer 6 seems to have the most diversity of pyramidal cell types. So I don't know if someday we could get to doing these experiments in human layer 6. Then we could probably come across a diversity of dendritic excitability patterns, I suppose, given that hopefully human layer 6 also has the most diversity of pyramidal cells, have very different projections to the rest of the brain.
Right. So there's developmental issue which is very, very intriguing. Right. So if you can divide the corticogenesis into two parts, in the human corticogenesis into two parts, you'll have the initial part, let's say, of the first half of gestation. What you'll get at that part is that the development of rodent and human brain or cortex is very similar in terms of the cells from the ventricular zone and the scaffolding that you get when the neuron migrate to the cortex and develop there. And so that's the first half. The second half, you'll see that there is some distinction between primates and rodents. And there are very unique aspects there in humans. For example, there is this discontinuity of the ventricular zone that is only relevant in humans and primates, and you do not see it in rodents. Like, you see the same areas of the ventricular zone, but what you don't see is this division, this discontinuity that happens in the second half of gestation. And in that part, there's all this massive activation of different genes and the proliferation of the cells is completely different. So the interesting part is that in development, the lower layers developed before the upper layers in the cortex. Mm-hmm. So what you get is that the layers 6 and 5 and maybe 4 are developing in stages which are more similar to rodents. Mm-hmm. And then when you have layer 2, 3, and layer 1, I don't know what to say about that, but layer 2, 3 in terms of excitatory cells, then that is more later in the development, and that has these unique aspects, right? Which are for humans and for primates, but not for rodents. So if you start from this, your null hypothesis would be, let's have a look at these upper layers, right? And we probably will find less dissimilarities in the lower layers. But that's not the end of the story. It doesn't mean that you don't want to go there. But in terms of development, right, why do you want to go to the upper layers? That's why.
Mm-hmm. Yeah. Cool. Mm-hmm. Any more questions? comments, thoughts, ideas?
Okay.
Yeah, I had just a bit technical. So if you can walk me through again how you separate the NMDA spikes and calcium spikes, how you're sure that you're blocking the NMDA input, not blocking, but separating that. And what's the percentage of this NMDA input and the calcium spikes that you see? It's a bit technical. I think I have the idea how it works. But since now we're like having a bit conversation out of the subject.
Yeah. So no NMDA spikes.
No NMDA spike.
No, because we don't activate synapses in order to get this spike. What we do is we inject current into the dendrite in order to get the spikes. Right? So not mediated by synapses.
Yeah, but then there is no activation of the synapses whatsoever. Nothing. So there's no side effect on that.
The activation. When is the activation of the synapse?
Like there's no side effect. So you're sure that there's no side effect, because I don't know. I've been having this conversation and I was told that it's possible to get the activation of the synapses. So if you're just stimulating the dendrite, then there will be some residual activation of the synapses.
Well, I cannot say that I see anything like that. Might be the case, but I do not see that. And the shape of the action potential, right?
Yeah.
Not similar to NMDA spikes in any way you think about it. Right? The properties not similar to NMDA spikes, and it's not similar to calcium spike as well. So you have different properties of these spikes. Right? As I told you, when you see it, it's relatively easy to distinguish. But if I can exclude some participation of channels which are sensitive to the input to the dendrite, to the stimulation to the dendrite, well, I cannot tell you from the top of my head that I can completely exclude that, but this is definitely would be a minor effect.
Yeah, because also I see that in the literature there are this DNA calcium, like the sodium calcium spikes that you mentioned. You don't have impact of sodium, but it seems that there's like big variability in different synapses that dendrites can function with. Right?
There is a big what?
Variability of the different dendritic spikes that were reported in the literature. Do you think it will converge to some particular type of dendritic spikes when the techniques will be more homogenized across laboratories?
Well, I think the opposite is true. I think it will diverge to more spikes, and I'll say that there is a reason why I think that, which I didn't show today, but there might be even more spikes than what we see. And just one thing, so you know, we did some NMDA spikes as well. So we looked at NMDA spikes, and these spikes in humans are really broad, broader than what you expect to see in rodents, what you see in rodents. So they are bigger and they are not mistakable. I mean, with what I showed you today, that's very clear. So, and you might have more than one type of NMDA spike in this cell. So you probably have more than what we know, not less, not converging, but diverging. Yeah, it's like now it's more particles in the human dendrites, in dendrites in general.